Fertilization is not a race. It is a highly selective process. Susan S. Suarez. We were told the fastest sperm wins, that fertilization is chaos, randomness, luck. That story is wrong because the uterus is not passive. It doesn't wait, it decides. Hundreds of millions of sperm enter the body. One reaches the egg. For decades, this looked like competition, like speed, like chance. But biology does not reward myths. It rewards filters. And the female reproductive system is one of the most selective environments in nature. A typical ejaculation contains 200 to 300 million sperm. Within minutes, most are already gone. Less than 1% pass the cervix. Only hundreds reach the fallopian tubes. Exactly one fertilizes the egg. This loss is not accidental. It is not passive. Most sperm are eliminated by female track processes. Selection begins immediately. The idea of a simple race has a history. Early reproductive biology was shaped by what could be seen. Under early microscopes, movement was visible. Sperm moved, eggs did not. So activity was assigned to sperm. Passivity to the egg. This visual bias shaped decades of theory. The sperm became the actor. The egg became the destination. The metaphor survived because it was intuitive, easy to explain, and easy to repeat, not because it was correct. When fertilization was later studied, at the molecular level, the narrative collapsed. The egg is not penetrated by force. It binds and it evaluates. It allows entry. Fertilization is not impact. It is interaction. The race metaphor remained because metaphors outlive evidence. To the female body, sperm are foreign cells. They carry paternal antigens. They are immunologically non-self. After intercourse, the uterus initiates a controlled inflammatory response. This response is measurable. Immune signaling genes activate. Blood vessels dilate. Leukocytes migrate into the uterine cavity. Neutrophils release extracellular traps that physically ensnare sperm. Macrophages ingest them. Millions are removed. Within hours, the majority of sperm are eliminated by post-mating immune responses. This is not pathology. It is regulation. Without this response, the uterus would be vulnerable to infection. Sperm clearance and immune defense are inseparable. The same mechanism that removes sperm also protects the female body. Selection and protection are the same process. This immune response is temporary. It peaks quickly, then resolves, strong enough to eliminate sperm, controlled enough to allow implantation later. The uterus is not hostile. It is selective. The cervix adds another filter. It is not an open passage. Cervical mucus forms microchannels with specific orientation and viscosity. Only sperm with correct morphology and strong forward motility can enter these channels. Abnormally shaped sperm fail to align. Sperm with weak propulsion are pushed backward. Around ovulation, the mucus changes. It becomes less acidic, more hydrated, less restrictive. But permissive does not mean neutral. The cervix still filters. It simply changes. Which sperm are allowed through? Beyond the uterus lies the uterotubal junction, a narrow anatomical bottleneck. Between uterus and fallopian tube, here selection becomes extreme. The opening is so restrictive that individual sperm often cannot pass alone. In animal models, sperm must cooperate. They cluster. They align their movement. They generate force together. Sperm cooperation is required to traverse the uterotubal junction if sperm numbers are too low. No amount of speed helps. Low sperm count does not just reduce probability. Sometimes it means zero access. Those that pass enter the fallopian tube. Here the female tract does something unexpected. It stores sperm. Sperm bind to the epithelial lining of the oviduct. They are held in place, alive, metabolically quiet, preserved. This reservoir can last for days, and it prevents premature activation. It synchronizes sperm availability. With ovulation, only a small number of sperm are released at a time. This matters. If too many sperm reach the egg, polyspermy would occur. Polyspermy is lethal. The female tract actively prevents it. release from the reservoir is not random. It is triggered by chemical signals associated with ovulation. Sperm are activated when in waves, not all at once. During this phase, sperm are guided. The egg and surrounding cells release chemical attractants. These molecules form gradients. Only sperm with the correct receptors respond. Only capacitated sperm can detect these signals. Capacitation itself is a selective bottleneck. Only a fraction of sperm ever reach this state. Another misconception is that all sperm compete equally. They do not. Some sperm fail mechanically, some fail chemically, some fail immunologically, others fail silently. For example, sperm with damaged DNA can appear normal. They can swim, they can move fast, but damage leaves traces. Membrane instability, oxidative stress, altered surface proteins. The female reproductive tract does not read DNA directly. It detects consequences of damage. Those sperm are less likely to survive immune exposure, less likely to bind to oviduct cells, less likely to respond to chemical cues. This is why sperm selected by the female tract show lower DNA fragmentation than unselected samples. Selection is indirect, but effective. Capacitation adds another filter. It is a biochemical transformation. Ion channels open. Membrane composition changes. Calcium signaling becomes possible. Without capacitation, fertilization is impossible. The female tract controls where and when this happens. Capacitation too early leads to failure. Capacitation too late leads to exclusion. Timing matters. This is why the reservoir exists. It is not a waiting room. It is a timing mechanism. Only sperm released close to ovulation retain fertilization capacity, even at the final step. Speed is not decisive at the egg surface. Binding is reversible. Weak binding fails. Only sustained molecular compatibility allows progression to fusion. The egg does not broadcast blindly. It emits signals that only some sperm can hear. The egg does not call everyone. It calls specific sperm. In 2020, a human study demonstrated something crucial. Egg-associated follicular fluid attracted some men's sperm more than others. The difference was consistent, repeatable, not random, not faster sperm, not stronger sperm, more compatible sperm. Eggs attract some sperm more than others. This attraction varies between women and between men. A woman's egg may chemically prefer a man who is not her chosen partner. This happens silently, without awareness. At the cellular level, genetic compatibility matters, especially immune system genes. From an evolutionary perspective, this makes sense. Offspring with diverse immune genes are more resilient. The female reproductive tract does not only filter for quality, it filters for compatibility. Selection continues at the egg itself. Fertilization requires a precise molecular interaction. Specific proteins on sperm must bind specific receptors on the egg. Without this molecular handshake, fusion cannot occur. The sperm does not force its way in. It is accepted. Once accepted, the egg changes immediately. Receptors are removed, the surface hardens. No other sperm can enter. One is chosen, all others are excluded. This is not chance, this is control. Fertilization is not chaos, it is not a lottery. It is a multi-layered negotiation between immunity, chemistry, mechanics, and evolution. The female body does not gamble with life. It filters for resilience. Charles Darwin described selection before mating. He never described what happens after. Now we know. Selection does not stop at attraction. It does not stop at sex. It continues at the cellular level. So ask yourself, if even life's beginning is curated, filtered, selected, how much of what we call chance was never random at all? See you.